Assalamu alaikum, my dear brothers and sisters. Welcome you all to my YouTube channel, Let's Learn Pharmacy. Yes, so today I am here to discuss about the fourth important question in pharmaceutical microbiology that belongs to 10 marks. That is, you observe here the second question. Draw a neat label diagram of a virus cell and explain the stages of replication. See here, this question is very important, my dear brothers and sisters. And this question is asked many times in 5 marks also, 10 marks also. But majorly I will give preference for this question for 10 marks. Okay, see a simple question. Here it was asked once in second question you are observing, and next it was asked in 5 marks here. The question number 8, write a note on not this cultivation. It is different. Yes, here see the 8th question, describe steps involved in the replication of the viruses. Same, both the questions are same. Answer is also same for them. See here, it belongs to unit 3 chapter 1. Here, before going to study that replication, we want to discuss what is this virus. Viruses are the smallest known infective agents. They are ultra microscopic and can only be viewed with electron microscope. They are minute infectious particles which can be visible through electron microscope, and they are obligate parasite. They live only in the living cells, means they multiply only on their living cells, means human host cell. Got it? And their genetic material is either DNA or RNA. Okay, both the genetic materials are not found in the viruses. Clear? Either they can be DNA or it can be RNA. Viruses are small, they are very small than the other organisms, and they are very small so that they can be transmitted through the bacterial filters. Got it? And this is the structure of a naked virus. Two types of virus, one is coated, another one is naked. It is a naked type of virus, okay, without any capsid or without any nucleocapsid, without any covering. Next we discuss about this is the capsid virus. These all are what you are observing, no? The pin shaped points outside. It is the envelope, my dear brothers and sisters. Okay, envelope it is. Envelope. Here also the envelope is covered. Capsid envelope. Got it? Now let's discuss. The main pattern starts from here. Replication of virus. What is this replication? Replication means simple dividing, my dear brothers and sisters. Replication is nothing but division. Division is known as replication. Now you tell me one thing. Due to the lack of the biosynthetic enzymes, viruses replicate by utilizing biochemical machinery of the host cell. What happen basically in viruses? No, viruses अपने आप में बायोसिंथेटिक एंजाइम प्रोड्यूस नहीं कर सकते. वो अपने आप में कुछ भी प्रकार का यूजफुल थिंग्स उन वायरसेस को दे नहीं सकते. वायरसेस, इसके कारण वायरसेस क्या करते हैं? दे एंटर इनटू द ह्यूमन बॉडी. ये वायरस है, ये ह्यूमन बॉडी के अंदर एंटर करते हैं, क्योंकि ह्यूमन बॉडी या एनी एनिमल बॉडी, प्लांट बॉडी, होस्ट बॉडी के अंदर क्या रहता है? उन्हें उधर जीव करने का एक गारंटी रहता है. ओके, दे कैन शेल्टर देयर. फॉर दैट्स रीजन दे आर taking the host body for their life. Okay, then the replication divided into 6 sequential stages. One is adsorption, penetration, uncoating, biosynthesis, viron assembly, release. One by one we will discuss in detail. Very important question, so don't miss this video. Share this video with your friends, and the link of the notes available in the description. You can go and check it out and write that notes. Please practice those. Practice. The first type of the synthesis, biosynthesis, the first type of replication is adsorption or attachment. It is the first event in any viral infection in which the virus comes in contact with the cells by random collision and gets attached to the cells. So for an example, see, this is the virus. What is this? It is the virus. Now this is the one human body or animal body. Okay, what happens? At the random collision with this human body, it gets attached into the human body. This is the concept. This adsorption is specific and depends on the surface of the receptor host cell. How the receptor host cell is, it depends on that. Okay, if it is hard, means it requires some of the specific attachments. Got it? Animal viruses have specialized attachment sites disturbed over the surface of the viron. Some of the animal viruses, they are having the specialized attachment sites, means they are having the cone shaped structure over their envelope. Okay, what is this? It is an envelope. They are having the cone shaped structure over their envelope, which can they easily sight, or they can be situated on the human or animal. Now the next is, some of the example: orthomyxoviruses and paramyxoviruses attached to the glycoprotein spikes. They having the glycoprotein spikes. These are two viruses. They attached by means of these glycoprotein spikes. In our football shoes, you are observing the spikes, no? That spike, the same. These are the same. When we are observing the under electron microscope, they looks like the same. But that way, they get attached. Where? To the host. Then the next one is penetration, means insertion. Penetration of the viruses occurs either by engulfment of the whole viruses or by fusion of the viral envelope with the cell membrane, allowing only the nucleocapsid of the virus to enter the cell. Simple. Means entering or penetration, my dear brothers and sisters, it can be penetration or going inside the whole virus either, or the virus sends only his nuclear envelope, nuclear capsid, or a nuclear genetic material into that host cell for the multiplication. Okay, there are two situations. The third one is uncoating. Here later I will discuss. One diagram is there. Here you can get all the things which you have discussed earlier. Uncoating, the process involves the physical separation of the nucleic acid from its outer structural component. Uncoating, my dear brothers and sisters, it is the process where it involves the physical separation of the nucleic acid. The main material in the virus is the nucleic acid. Nucleic acid only produces the replication. Okay, it only helps to promote the replication. Okay, what happens? These will uncoat the whole other materials except that nucleic acid. Okay, they will remove the nucleic acid. Now, it is assumed that host components and proteolytic enzymes within the lysosomes cause this uncoating process. Who causing this uncoating process? Uncoating is caused by the host only, host components like proteolytic enzymes within the lysosome. These proteolytic enzymes are the acidic enzymes, protein enzymes. These cause those viruses to uncoat or to shed their coverings, to remove their plastic. Which one? The proteolytic enzymes. Now, in adenovirus and some of the parvoviruses, uncoating is accomplished or maintained by the cellular proteases present in the cytosol. Means in simple, cytoplasm helps for the uncoating process, where in these only two, that is one of the adenovirus and parvoviruses. This is the three type of the replication process of the virus. Now let's discuss the fourth one, that is biosynthesis. I have told you the virus can't be biosynthesis, or they can't synthesize anything by itself. Either they will have to enter into the living body for their biosynthesis, or any other living body, because they itself can't biosynthesize. Okay, so they want any other living body. After uncoating, you have seen the process, the third one, that is uncoating process, shedding process, removing the covering membrane. After uncoating, what happened? Yes, the next one is in general, the nucleic acid genome of the most of the DNA viruses are synthesized in the host nucleus. I have told you, my dear brothers and sisters, the nucleic acid means the genome of most of the DNA viruses are synthesized in the host. Basically, the host nuclear cell helps to give biosynthesis for the viruses. They give food, they give shelter, everything is given by the host only. For an example, if the virus, coronavirus or Omicron virus, if it enters into the human body, means the whole process, the whole biosynthesis, the shelter is given by the human body only for those. They're just the work of those viruses is they are just living. They are eating, everything they are doing. वो बोलते हैं ना कामचोर, सभी खाते हैं, सभी पीते हैं, सब हमारा ही जिस्म इस्तेमाल करते हैं वो, ओके। और जाते वक्त जब वो बॉडी को छोड़ के जाते हैं ना, तभी उस समय सभी लेके जाते हैं, लेकिन हमको कमज़ोर करके जाते हैं। देखिए निमोनिया, सर्दी, जुकाम, खांसी, लंग कैंसर, लंग डैमेज करते जाने का। दैट्स द प्रॉब्लम ऑफ दिस DNA वायरसेस। हाँ, नाउ एक्सेप्शन ऑफ पॉक्स वायरस सिंथेसिस ऑल ओवर द कंपोनेंट्स इन द साइटोप्लाज्म। देखिए मोस्ट ऑफ द वायरस जो है ना, एवरी वायरस इज सिंथेसाइज इन द न्यूक्लिक एसिड ऑफ द ह्यूमन DNA, बट दीज सम द पॉक्स वायरस ये मेनली फॉक्स के अंदर, फॉक्स या शीप्स के अंदर ये वायरस पाया जाता है दोस्तों, ओके। ये वायरस है ना, पॉक्स वायरस, दे गेट सिंथेसाइज इन द साइटोप्लाज्म और दे बायोसिंथेसाइज इन द साइटोप्लाज्म। हाँ, initially viral DNA is released into the nucleus of the host cell to transcribed into the mRNA, then translated to perform new viral protein. देखिए this viral DNA, it is released into the nucleus of the host cell. For better understanding, see here, what is this? This is the nucleus, okay, nucleus. For just example I am telling, initially this viral DNA, if it enters into this, what happens? It will release into the nucleus of the host cell this RNA. Sorry, not viral DNA. It release where? It release into the host nucleus, host DNA, host cell. Then it get transferred into the mRNA. You studied in your second sem of the B pharmacy in biochemistry that synthesis of new protein from the mRNA transcription, then it transfer to the RNA, then RNA to the means tRNA transfers RNA, mRNA, then it transfer a new protein is formed, okay. This formation of the protein then translated to form a new viral protein, okay. New viral protein is formed by the viral DNA. किसके अंदर? देखिए आपको समझाता हूँ मैं, आप नहीं समझे होंगे शायद। दिस इज द न्यूक्लियस, हाँ न्यूक्लियस, दिस इज द DNA वायरल। अब DNA वायरल जो है ना, ये किसके अंदर जाता है? न्यूक्लियस ऑफ द होस्ट सेल के अंदर जाता है। जाके यही DNA वायरल DNA इट स्टार्ट्स इनटू mRNA, मैसेंजर RNA, देन इट ट्रांसफर्स, दैट इज ए tRNA के बाद में ये अलग ही तरह का एक प्रोटीन फॉर्म करता है, दैट इज ए पेप्टाइड चेन इज फॉर्म, वायरल प्रोटीन फॉर्म हो जाता है दोस्तों, खत्म, ओके। दिस इज द procedure. The early viral proteins are concerned with the replication of the viral DNA. यही viral DNA, okay, protein only viral DNA, ये replicate होता है, ये अदर्स फॉर्म के अंदर replication होके एक long thread of protein फॉर्म हो जाता है दोस्तों, समझ लीजिए। सेम ये जो वायरल DNA का प्रोसेस सेम रहता है, किस तरह? द प्रोसेस इज सेम लाइक द ह्यूमन DNA. How the human DNA is get replicated? Same in the how the replication of the DNA is by means of the transcription, translation. Same way this viral DNA is getting replicated. That's why our body cannot define this type of viral DNA. यही एक वजह है दोस्तों। अभी देखिए HIV वायरस हो जाता है, HIV वायरस बेसिकली वायरल DNA कन्वर्ट होके बॉडी के अंदर इनके रेप्लिकेशन इतने हो जाते हैं, हमारे बॉडी से ज़्यादा अगर ये वायरल DNA हो गए तो तब हमारी बॉडी कमज़ोर पड़ जाती है। आवर बॉडी गेट डिस्ट्रॉयड, आवर बॉडी गेट्स न्यूट्रलाइज्ड, ओके। व्हाई? बिकॉज ऑफ द सिंथेसिस ऑफ दिस वायरल DNA. हाँ, दिस इज द कॉन्सेप्ट. In case of some RNA viruses, example picorna virus stays in the cytoplasm where it mediates its own replication and translation into viral proteins. Some of the viruses like picorna viruses, these viruses they get replicated into cytoplasm and they transfer the viral protein chain. Now my dear brothers and sisters, it is the last viron assembly. Assemble means joining, assembly virals, that assembly of the various viral components into the complete virus particle occurs shortly after replication of a viral nucleic acid and may takes place in the cytoplasm or nucleus. Means these assemble or these joining. Assemble मतलब एक सोल्जर की तरह एक लाइन बनाना, assembly होना। अब प्रेयर्स के पास हम कैसा assemble होते हैं ना, सब मिलकर, हाँ उसी तरह assemble होना। ये assemble कभी होता है जब all the viral components complete their virus particles वायरस प्रोटीन चेन जब बनाते हैं तब दे असेंबल होते हैं, आइदर दे कैन असेंबल इन द साइटोप्लाज्म और इन द न्यूक्लियस. द कैप्सोमियर प्रोटीन्स एनक्लोज द न्यूक्लिक एसिड टू फॉर्म द वायरल न्यूक्लियोकैप्सिड. ये जो कैप्सिड रहता है ना, कवरिंग मेम्ब्रेन. नाउ द लास्ट इज रिलीज प्रोसेस. कैसा ये रिलीज होते हैं? इट इज द फाइनल इवेंट इन वायरल रेप्लिकेशन. इट रिजल्ट्स इन द रिलीज ऑफ द मैच्योर वायरॉन्स फ्रॉम देयर होस्ट सेल. देखिए अभी ये एक सेल है, होस्ट सेल के अंदर ये सब कुछ कर रहे हैं, ये डेवलप हो रहे हैं, ये खाना खा रहे हैं, रेप्लिकेशन हो रहे हैं. आफ्टर रेप्लिकेशन असेंबल भी हो गए हैं. असेंबल होने के बाद मेरे दोस्तों दे गेट रिलीज होने के बाद ही ये काम करते हैं. ओके, बगैर रिलीज के इनका काम हरगिज नहीं. आफ्टर द रिलीज ओनली दे शोज देयर एक्शन ऑन द ह्यूमन बॉडी. अभी जुकाम या सर्दी वगैरह एलर्जी कैसा होता है? इफ द वायरस इज कंटेन, इफ द वायरस रिलीज इन द ह्यूमन बॉडी, एट दैट टाइम ओनली नो दीज ऑल सिम्टम्स वी ऑब्जर्व इन योर बॉडी. वायरस मैचुरेशन एंड रिलीज अकर्स ओवर कंसिडरेबल पीरियड ऑफ टाइम. ओके, इनको एक पीरियड होना है. दे कंटेन सम पीरियड. Okay, in a considerable period of time they get released. Naked viruses may be lysis of the host cell without damaging the host cell, exception of the poliovirus. देखिए जो वायरस नेकेड है, नेकेड वायरस मीन्स दे कैनॉट एनक्लोज द मेम्ब्रेन. कैप्सिड जो वायरस पर नहीं रहता है वो इतने डैमेज नहीं होते दोस्तों, और वो ब्रेक डाउन होकर सेल से बाहर निकल जाते हैं. पोलियो वायरस ऐसा वायरस है, ये डैमेज करता है होस्ट सेल को. जब बाहर जाता है ये डैमेज करता है. मैं ये नहीं कह रहा हूँ कि सभी वायरसेस डैमेज करते हैं बॉडी को. नो 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 नो. जो वायरसेस नेक्ड रहते हैं वो डैमेज नहीं करते हैं, एक्सेप्ट पोलियो वायरस. पोलियो वायरस. In case of enveloped viruses, the nucleopeptide acquires its final envelope from the nuclear or cell membrane by budding off process before release out of the host cell. Okay. एनवेलप वायरस के अंदर क्या रहता है? ये बडिंग फॉर्म करते हैं. बड फॉर्म करके अपने आप पर चारों तरफ गहराई में दे डेवलप्ड दे स्पाइक्स. ओके, फुटबॉल शूज स्पाइक्स जो रहते हैं ना, उसी तरह चारों और इनके अतराफ में ये स्पाइक्स डेवलप करते हैं. ओके, स्पाइक्स द कोन शेप्ड स्ट्रक्चर डेवलप करके बाहर निकल जाते हैं, क्योंकि और दूसरों को अटैक भी करना है ना. हाँ, इसलिए आपको मैं इन सिंपल वे इधर भाई डायग्राम में बता देता हूँ. इट इज़ द रेप्लिकेशन ऑफ वायरसेस. देखिए फर्स्ट वन ये वायरस है. ओके, मैच्योर वायरस. इट गेट अटैच. ये अटैचमेंट, इट इज़ द फर्स्ट रेप्लिकेशन स्टेज. अटैचमेंट, कोटेड पिट. अटैचमेंट के बाद पेनेट्रेशन, इंसर्शन ऑफ द वायरस इनटू द होस्ट सेल. ओके, कोटेड वेसल. देन एंडोसोम. आफ्टर दैट अनकोटिंग. मतलब ये जो कैप्सोमियर्स कैप्सूल रहते हैं ना, दे गेट ऑल रिलीज्ड and means they get removed from this virus and uncoating virus, and these material DNA, viral DNA or viral RNA, it transferred into the capsid assemble. They get assembled in the line. After the assembly, what happens? Nucleocapsid assembles envelope proteins. They form the envelope protein. After that, budding process. Okay, it is the budding process. आपको बताया था मैं, budding is the process where the capsid get developed. After that the process of maturation and release of the virus. A mature virus get released. Okay, insertion of the envelope proteins, then glyco isolation of the envelope protein is completed. ये सभी प्रोसेस है दोस्तों. ओके, अभी एनवेलप देखिए. एनवेलप इट इज कंपोज ऑफ द ग्लाइकोप्रोटीन. दिस इज ओनली ग्लाइको आइसोलेशन ऑफ द एनवेलप प्रोटीन इज कंप्लीटेड. ओके, ग्लाइको आइसोलेशन. ये सभी आप एक बार देख लीजिए, सिंपल याद रखिए दोस्तों. फर्स्ट वन इज अटैचमेंट, पेनेट्रेशन, अनकोटिंग, असेंबल, उसके बाद मैचुरेशन एंड रिलीज ऑफ द वायरल डीएनए और वायरल आरएनए. Okay, one point you remember, the virus cannot be contained both RNA or DNA as a genetic material. Either they having or they contain the DNA as their genetic material or RNA as their genetic material. This is the concept you have to remember. Yes, my dear brothers and sisters, thank you for supporting me. And if you like this video, please subscribe my channel, Let's Join Pharmacy, and share this video with your friends, cousins. Thank you. Assalamu alaikum.